Okay, Assalamualaikum. For S9, S10 and S25, I would like to explain to you on your second experiment, which is on kidney function and physiology. So this is supposedly the second week for you to do this experiment. So this is week 6, okay? So basically for this experiment, the objective of this experiment is to study the structure and functions of the excretory system, which are essential in excreting waste and osmoregulations in humans. And second objective is to measure the effects in the consumption of various concentration of liquid towards urine output. Okay. So this is the two objectives of this experiment that you must include when writing your lab report. Okay. And then for this experiment, the first part of this experiment, which is 2.1, which is physiology and anatomy of the kidney. For this part of the experiment, you are going to observe model of the human kidney, model of the urinary system, and human kidney. Okay, it is prepared slide of human kidney. So the apparatus that you will need is light microscope. But because this experiment is not face-to-face, you are not going to use the light microscope. You are just going to observe and draw the image of the prepared slide of the human kidney. So there are structures that you have to draw and also label. I will show you later the picture for all the three items, okay, materials. Basically, the procedure: observe, draw and label the structures of human kidney and the urinary system. So I will show you the picture later. And then if this experiment was to be done face to face, you will need light microscope and you have to observe the prepared slide. Okay, so for this one, you are just going to observe the image, okay, for the observation of the prepared slide of human kidney. And then for the second part of this experiment is on urinalysis. Okay, so before I go into this the explanation of the second part of the experiment, I will have to show you the image, okay, for the three items. Basically, what you have to do is that you have to make sure you download the manual, okay, the lab manual, the soft copy of the lab manual in the UFuture. So when you have to enter into your UFuture account, and then you have to go to the folder prepared by Sir Rahimi. Okay, Sir Rahimi prepared the folder. So make sure when you enter into the folder, you download this folder, okay, this file, okay, which is experiment 2, kidney function and physiology. So download, okay. So in this file, okay, you will have, so download, okay, open. Okay, so this is the image, okay, of the model. Okay, so you have to observe this model and draw. So remember, every time when you draw your drawings, okay, or any diagram, you have to draw using pencil. And then another thing that you have to note is that whenever you draw any diagram, especially model of anything, you have to draw on a piece of plain A4 paper. Okay, you have to draw it large. Draw it using pencil. Draw it large on a piece of plain A4 paper, and then make sure you label all the organs and structures involved. Okay, so you have the kidneys. Okay, above here you have the adrenal gland, and you have the ureter. Okay, and then you have the blood vessels. Okay, the aorta and also the veins. Okay, so make sure that you draw the model correctly and label all the structures. Okay, so all the structures will have marks for each of the structure that you label. So make sure you label correctly all the structures and organ involved. So this is the urinary bladder, as you can see. Okay. So the second model that you have to draw, and make sure that when you draw your model, make sure you label. Okay, you label it. So this is model of the urinary system. If you do not label your drawing or your diagram, so marks will be deducted. Okay, so make sure you draw it neatly, label all the structures, and label also the Venn diagram here. So the second diagram here is model of the human kidney. So here is the human kidney. So make sure you draw it neatly using pencil and label all the structures involved, the nephrons, okay, the renal medulla, the renal cortex. This is the adrenal gland above the kidney, okay, the blood vessels. So basically, in order for you to help you label all the structures, you can refer to your books, and also you must watch the video that is prepared by Dr. Khaniza explaining on this experiment. I will give you the link, okay, in the description below. Okay, so make sure you click on the link to watch the video done by Dr. Khaniza to explain in detail, okay, on the two models and also the image below here. Okay, so make sure you watch the video prepared by Dr. Khaniza. So second is the third image, okay, that you have to observe is the microscope observation of the human kidney. So this is the prepared slide that is observed under 100 times magnification. So make sure when you draw this, you draw it big and large. Okay, so as you can see, so this is the cross section of a human kidney. As you can see the glomerulus, the Bowman capsule, okay, and around it you have the proximal tubule. Okay, so once you have downloaded this file again, so you can enlarge the image, okay, to help you observe better lah, okay, and also to help you draw the image correctly. So you have to draw really large, okay, a maximum at least, okay, half a page, okay, at least half a page. So draw using pencil, label all the structures, which one is the Bowman capsule, which one is the glomerulus, okay, the proximal tubule around it, okay, and then you can also see the blood vessels, okay. And then so this is the image observed under 100 magnification. So this is under 400 times magnification. So you can really clearly see the image, okay. So this is basically this is the glomerulus, okay, and this is the Bowman capsule. You have the proximal tubule. You have the blood vessels surrounding the nephron. Okay, so this is part of a nephron basically. So make sure you label correctly. Make sure you listen to the explanation given by Dr. Khaniza. She explained to you in detail the structure involved in this slide. Okay, so draw and label all the structures. So make sure not to forget the main title for each of the image or drawing that you've drawn. Okay, so this is the one that you have to download for you to refer for your observation for your drawing, okay, in the result part of your lab report. And then for the next one, okay, is the, okay, I will go to the next part, okay, of the experiment, which is 2.2 on urinalysis test. So for this one, it involves urine collection and consumption of different concentration of liquid. So you will need, supposedly, okay, you will need 3 volunteers, okay, 3 volunteers. If this experiment was to be done face to face in the lab, there should be 3 volunteers, okay, that will drink. The first volunteer that will drink 800 ml of water. The second volunteer will drink 800 ml of 0.9% diluted salt solution, okay. And then the third volunteer will drink very concentrated salt solution, which is 80 ml of 9% salt solution. And then in order for you to prepare these two salt solution, you will need a measuring cylinder, urine container, and also gloves, because you are dealing with urine, so you have to really take care of the hygiene here. Okay, and then so this is the procedures. So each group must have at least 3 participants with no health issues regarding kidney and heart. Okay, each of the participants must first empty their bladder and measure the volume, transfer some urine into a urine container. So this is the initial urine. Okay, so this is the initial urine before you drink all the 3 solution here. Okay, the 3 solution. So take your sample, your urine sample, and measure the amount of urine you produced, and also observe the color. Okay, the initial color of the urine, to compare with the urine after you have drink these 3 solution. Okay, so note, collect urine sample midstream inside a urine container. So this is to avoid contamination from the external genitalia, and to collect urine that has been cleansed of pus cells, and also bacteria normally found in the urethra. So that's why you have to collect the midstream, okay, to avoid the contamination in the at the beginning of the urine. Okay, so this is the 3 solution. So each of the participant is required to drink the following liquids, okay, which is participant A, 800 ml of water, participant B is 800 ml of 0.9% of sodium chloride, and participant C is 80 ml of 9% sodium chloride. So to prepare this solution is by using this formula. So you just use this formula to prepare 800 ml of 0.9% sodium chloride, dissolve 7.2 gram of salt, okay, in 800 ml of drinking water. And then for 80 ml of 9% salt solution, so dissolve 7.2 gram of salt in 80 ml of drinking water. Okay, so this is how you prepare the 2 salt solution. So after you have drink after each of the participants have drink the respective liquids, okay, so you have to wait for 60 minutes. So make sure within this 60 minutes you do not drink any other fluids or eat. Okay, so this will influence the final urine collection later. So make sure you do not drink or eat within this time range. Okay, so each participant will re-empty their bladder and measure the urine volume, and some are transferred to a urine container. Okay, so this urine is the urine after 60 minutes. So if the samples are not collected within the 60 minutes, make sure you record the exact time. Okay, so so this is the instruction if it were to be done face to face. But for your lab report, you must come out, okay, you must come out with the expected result. You must you must write a hypothetical result, okay, a hypothetical result of the urine, the amount of urine for the initial urine for the 3 participants, okay, and also the amount of urine for the final urine collected for the 3 participants. So for example, if participant A were to drink 800 ml of water, so obviously the amount of urine produced for the final urine collection will be a lot more, okay, a lot more compared to the initial urine. So you have to come out with your hypothetical and logical result. Okay, if you do not want to come up with your hypothetical results, then you could, okay, you could discuss among your team member, okay, to do the actual experiment. Okay, so if you have access to salt, for example, you could prepare your salt solution and drink the salt solution. Okay, so you could do the experiment at home, okay, and gather the results and put your results in a table form to compare the initial urine, the initial volume, the volume of the initial urine, and also the volume of the final urine collected after 60 minutes. Okay, so you could also observe the difference in the color of the urine before and after. Okay, you could observe the color and the transparency of the urine sample for the initial urine sample and also for the final urine sample. Okay, so in your table you could include one, obviously the volume. Okay, the volume for the initial and the final urine collected. And then another result that you could observe is the color of the urine. Okay, the initial and the final urine collected. And also you could observe the transparency of the urine sample collected. Okay, so those are the results that you could include in your results. Okay, so make sure that when you record your results, it must be in a table form and label the table. For example, table 1. Okay, table 1. What is the title? So you give the title for that table. Okay, and then obviously your lab report must, after results, okay, you must include discussion. So in the discussion part, I want you to discuss on the function of the kidney. Okay, because you have observed and draw the kidney. Okay, the kidney, the model of the kidney. So you have to draw. You have to discuss the function of the kidney, the function of the urinary system, and also the function of the nephron. Okay, in osmoregulation and also in the formation of urine. And then for the discussion for the second part, I want you to discuss your results on the experiment 2.2. Okay, discuss your results on experiment 2.2. It means that you have to discuss what is the effects of drinking too much water and drinking concentrated water towards the formation of the urine. Okay, so how does your nephrons, okay, involved in the formation of urine for the 3 different solution. Okay, so you have basically for this discussion, you have to discuss your results. Okay, you have to discuss your results based on the theory. Okay, based on facts. Okay, so and then last of the discussion, you have to conclude your lab report. Okay, conclude your experiment. So conclusion must be based on the initial objective. So did you manage to achieve this objective? Okay, so for conclusion, it does not have to be long. Okay, it is about 1 paragraph, something like that. Okay, and then or half a page. So do not write a conclusion too long. And then after conclusion is discussion. Is references. So reference, you must at least include 2 or 3 references, either from books or from the internet. So this is that's the end of the explanation for my explanation for this experiment. So I just would like to highlight for S9 and S10, you must submit your lab report in the drive that I have shared. Okay, okay, which is in this folder. Okay, so you have to submit your lab report on the 11th of February. Okay, so I give you 1 week for you to do the experiment and to complete the lab report. So make sure you submit in this folder that I have shared with the head of the team. Okay, and then for S24, because next week on Friday is holiday, if you refer to your lab schedule that you can download in the future. So next week on Friday, it is Chinese New Year. So you must submit also on the 11th of February, okay, in the drive that I have shared. Okay, so that's the end of my explanation. So again, I would like to remind you to watch the video prepared by Dr. Khaniza, that you can watch the video in the acid biology YouTube channel. Okay, so watch this video. Okay, I will give the link in the description below. So you just click on the link, and then make sure that after you have watched this video and you have watched the video prepared by Dr. Khaniza, you fill up the Google Form attendance. Okay, that's it. Thank you, class.